Hi to everyone who's just logging in now. Welcome to Exploring Science. We're just getting some people registered into the event, but while you wait, we have a little activity. And this activity is from last week. We asked you to take or draw a picture of an animal that you can see outside, maybe when you go for one of your walks during the day, or you get to see it out of your window. So if you want to either hold up one of your pictures that you found, or you can type it into the chat, the name of the animal that you found. And then we asked you to see if you could find a cool fact about that animal. So I'll start with mine, but if you want to put your answers in the chat, and then maybe we can all share what we found. So I found this American robin in my front yard. So I'd seen this bird a lot, but I didn't actually know what it was. So I looked it up by its red or orange chest and its yellow beak. And it's something that's called an American robin. And it turns out that it's the state bird of Connecticut. So it's a bird you might see around all parts of Connecticut. And it's also the state bird of 2 other states. So if you want to guess in the chat what other states you think might have the same state bird as Connecticut, you can put your answer in there. Awesome. So the other 2 states that have the same state bird as Connecticut are Michigan and Wisconsin. So those are really good guesses. Someone actually got Wisconsin too. So I also found a little game on Twitter, and it's also on YouTube, posted by the tag is Afro Herper. She's a herpetologist, so she studies reptiles and lizards. And every week she posts a game called Find That Lizard. And she posts pictures just like this. And then you have to see if you can find that lizard. So if you want to see if you can look around this picture and see if you can find a lizard in this picture. And if you want to do it every week, you can follow her on Twitter or her YouTube, and you can play every week. So I put 3 boxes of places where I thought the lizard might be. So if you want to guess whether you think it's in the yellow box, the blue box, or the red box. All right, awesome. Yeah, so you were right. It was in the red box. So I circled it here, but then there's a blown up picture of what the lizard looks like. So if you follow this game, she also gives you fun facts about lizards, so you can learn more about the lizards that you're looking for and the lizard that you find. So this lizard actually had its tail kind of truncated or cut off, so a little bit of its tail had been missing, but lizards can grow back their tails. So it looks like we've got almost everyone logged in here, so we'll go ahead and get started with our event for today. So welcome to Exploring Science. Thank you all for joining us. We're going to take a deeper dive into the world of science. And this is an event brought to you by Open Labs and Flip Science Fair. So if you've ever been to the Flip Science Fair or the Science Cafe, this is kind of a virtual merger of those 2 events. And today, our first speaker is going to be Rick, and he's going to be presenting his poster that will teach us about chemicals in the brain and how we learn. And our second speaker will be Chris, and he's a graduate student in pathology who will be talking about stem cells and how we can use them to build new organs. Thank you all for joining, and we hope you have a great time at our event.
So good afternoon, everyone. My name is Rick, and I'm a 5th-year PhD student in the neuroscience program here at Yale. And I'm super excited to talk to you today about how I've been using milkshakes, mice, and lasers to study learning and memory. But first, I want to give you a little bit of background about me and how I got into science. I'm originally from a small town in North Florida, and when I was growing up, I didn't know what it meant to become a scientist or how to go about becoming one. Part of that is because I was the first in my family to go to college, so I didn't really have any guidance on how to get there. Another part is that I didn't see many scientists that looked like me. And then a final part is that my family didn't have a lot of money growing up. So I had to work really hard in high school to get scholarships and grants so that I could go to college. And eventually I got to go to the University of South Florida. And I was really excited to learn more about science there, because I found out that doing real science isn't just memorizing stuff out of textbooks. You got to work with your hands in the lab, design really cool experiments, and discover the things that are written about in textbooks and that change the way that we view the world. So after that, I was hooked, and now I'm here. And the rest is not quite science, but more so history. And I wanted to share that with you because it's important to let you all potential future scientists know that you don't have to be from a family with a lot of money, look like the majority of current scientists, or have parents that are scientists to become a scientist now. So if I can do it, you can definitely do it. All right, now let's dive into some of the science. My background is in biology. So when I approach neuroscience, I'm really interested in what's going on inside the brain at a cellular level to cause all the complex behaviors around what we do. So our actions, emotions, or even more abstract things like how we learn and store memories. In particular, I'm interested in why we learn about some things faster than others and what makes some memories stronger than others. And by stronger, I mean harder to forget or something like that. Now, I find this really interesting by itself, but it's really important to study this as well, because if we can help, because if we understand the building blocks of how learning and memory work normally, we can help people who have disorders around memory, such as Alzheimer's disease, which is an example of where people have a hard time remembering, or addiction and PTSD, which is sort of the flip side of that, to where memories are too strong and they affect people's lives negatively. Okay, now with that, I have to explain to you 3 pieces of background information in order to get you to make sure we're on the same page when discussing my experiments. So the first is the brain area that we're going to look at, and that's the basolateral amygdala, or BLA. So the BLA is a brain area important for positive and negative memories. And by important here, I mean something really positive or something really negative. So your brain is going to be active during that experience. And also, if you recall that experience later, it'll be active as well. The second thing that I want to talk to you about is acetylcholine. So this is a neurotransmitter or a brain chemical. So let's say it together, acetylcholine. So it's really not important if you know how to say it or spell it. I forget how to spell it all the time. But for the rest of the talk, if you see this symbol, I'm talking about the neurotransmitter acetylcholine. And the reason why I'm so interested in this is because it serves as sort of a traffic light for the rest of the brain. And that's really important because we have a lot of brain cells. Does anybody know how many brain cells we have? Go ahead and type it in the chat if you have an idea. Trillion, 100,000, okay. Uncountable, a lot, a 1 with a lot of zeros. Awesome, so there are a lot of really great answers. And the real answer for an average human is around 86 billion. And that's a billion with a B. So a lot is a ton, exactly. And you can imagine that if you had that many things going on in your brain at once, it might be a little crazy for you. So it could, for instance, happen to where you might have a seizure if all of your brain cells are active at the same time. So acetylcholine is able to cause things to slow down if they're not needed and things to speed up if they are needed, depending upon what your brain needs to do. And so these 2 things together are some work that I'm going to discuss from my colleagues in New York that did some cool experiments that involve mice and negative memories. But first, I want to take a second to talk about animal research, because working with animals is a big deal. Not only is it a big responsibility, but it's a privilege, and one that is not taken lightly. All of the experiments that we're going to talk about had to be approved by a group of veterinarians to make sure what we're doing is as humane as possible. They could not be done without using other animals, and will provide important data to move science forward. I, as well as all the other scientists I know, take this responsibility very seriously and don't take for granted the sacrifice that these animals are making. All right, so bringing those 2 things together, the brain area and the acetylcholine, some of my friends in New York showed that for mice that were learning about a negative experience, if there was more acetylcholine in the BLA at the time of learning, it made there be a stronger memory, which means it was harder to forget. And so I thought this research was really, really cool. And this led me to my hypothesis for my research, because remember, the amygdala, the BLA, is important for both positive and negative memories. So my hypothesis was in 2 parts. I thought that if we make there be more acetylcholine in the BLA during reward learning, it will make mice learn faster and have stronger memories, okay? But what I'm really excited to talk to you about is how we're actually increasing the acetylcholine levels in the BLA. And I'm going to introduce you to an idea called optogenetics. So optogenetics is just a big, fancy, mashed up word for us to say light plus genetic engineering and how we're using that to activate certain brain cells with a laser. So this is a cartoon of a brain cell. So this is its cell body, and that's where it mostly hangs out and lives. And then it sends its arm out to release acetylcholine. And so that's normally happening in the brain all the time. But for the opto cells, what we're doing is that we're changing them so that they are now glowing green, so we can see which ones we're targeting. And also, so now whenever we shine a laser at them, they release more acetylcholine than normal, because neurons usually don't do this. So this is a really great way for us to increase acetylcholine levels in the brain. And let's see what this looks like in a real brain. So this is a mouse's brain. If we were to cut sort of this direction, moving backwards through my forehead, sort of, maybe about 30% of the way. And all this green is where I delivered the opto tool. All right. And this is a zoomed in image here of where those cells live, which one part of the brain where they live a lot is the nucleus basalis. And so just to make this clear, I'm not actually adding brain cells, but I'm just changing the ones that are there, and specifically the ones that release acetylcholine. All right, so if they live here, they're going to be sending their arms to release acetylcholine in the BLA. And so you can see this outline here that I drew. It looks kind of like an almond, and that outline is right here too. And can you see all that green spaghetti looking stuff? That is the neurons that live in the NB that are sending their arms out in order to release acetylcholine. And this black area is actually where the optic fiber was. So an optic fiber is just kind of like a flashlight that allows me to point the laser where I want to specifically. And this is going to cause the neurons that live here to release more acetylcholine. All right, so now we're going to bring all those things together so that we can talk about these experiments. All right, I'm going to close something down real quick. Okay. Now, what we have is I injected the opto tool into the NB so that I can make all of these acetylcholine releasing neurons glow green and also respond to laser. But for the BLA, I'm implanting an optic fiber above it, so I can turn on only these brain cells, making them release more acetylcholine. And I get to do this in mice who are awake and learning about it, a rewarding task. And the task they're learning about is this. So I put them in a box for 30 minutes a day, and I'm training them to where whenever they hear a sound, the sound, it sounds like this, and the mouse has to put its nose in a hole. And if it does that, then it's going to get a milkshake. And mice actually really, really love milkshakes a lot more than cheese. So they're very motivated to do this. But this is kind of tricky because I can't just tell the mouse what I want it to do. It has to learn by trial and error because I don't speak mouse and they don't speak human. Okay? And so now what we're going to do is look to see if some mice learn faster than others. And we're going to compare these two groups, the control mice, to where the neurons are releasing a small amount of acetylcholine like normal, or the opto mice, which have the brain cells that are going to release more acetylcholine whenever we stimulate with laser. So let's look at this data first. In this learning paradigm or learning task, if they hear the sound, they're supposed to nose poke and they get milkshake. Okay. Now, each day here on the x-axis is 30 minutes a day that they went into the box. The y-axis is the number of milkshakes that they got. This green dotted line is when the data points go above that, it means that they've learned. And the two groups here are control and opto. So each data point is the average of 6 of those mice in each group. So first, let's look at what the control mice do. How long does it take them to learn? So it seems like they cross the green line for sure at about day 8. But now I want to ask you, how long does it take the opto mice to learn this task, would you say? So write down in the chat what you think. So how many days? Does it look like it's day 1 they learn, day 3, 4, 6, 12? Wow, I've seen a lot of 4 days. Wow, people are spot on. Day 7, okay. 6, 3.5, awesome. Those are all great guesses. And you're right. It was about day 4 or so that the mice were able to, the opto mice learned. And so does that mean that these mice are learning faster or slower than the controls? Faster. Okay, some people are saying faster. Right, exactly. Because it took them half as many days to learn. And remember, it's not the laser that's doing this really. It's the fact that we're increasing the amount of acetylcholine in the BLA when they're learning. But one person might think, okay, well, maybe these mice are just sort of the lasers making them nose poke a lot, right? So what we did also was look at the number of errors they make. So if the sound is not on, but they nose poke, we consider that an error. And so you can see that they make a lot of errors at first, but the opto group actually decreases their errors faster than control. So they're really learning about what it is they're supposed to be doing really well. So that's the first part of our hypothesis, right? We showed that more acetylcholine in the BLA made mice learn faster. But the second part is, what about for the strength of the memory? So now we're looking at how fast they unlearn. So now if the sound comes on and they nose poke, they don't get any milkshake. And we want to see how quickly do they stop nose poking, because they're not going to get any milkshakes. And we see that both groups decrease their responding or their nose poking at about the same rate. And they know that they're not getting any milkshakes because they're really not making any errors after a phone. Okay, so that was the second part of our hypothesis, to where it didn't seem like we increased the strength of memory, but we did make them learn faster. And that's what I want to recap here. So more acetylcholine in the BLA during learning led to faster reward learning. So the mice learned about this association that the sound is supposed to, that when the sound comes on, they're supposed to nose poke, and then they get a milkshake. They learned about that faster. But more acetylcholine in the BLA during reward learning did not form stronger memories. So it's really interesting here how we saw different results. For future directions, what I'm interested in doing is actually measuring when acetylcholine is normally released in the BLA. So I get to use another technique that uses light, but in the opposite direction. So there's a really cool way to measure the amount of acetylcholine by making these neurons in the BLA glow green, and they glow sort of a faint green normally. But when there's a lot of acetylcholine in there, they actually glow an even brighter green. And so we can tell that there's been more acetylcholine released in there. And so this tells us a lot about what's going on normally, as opposed to us just sort of turning on the light switch and making there be more acetylcholine. And I think most importantly, I'm really interested in why is the BLA acetylcholine different for positive and negative learning? Because this is the same brain area, the same brain chemical, but for different types of learning, it affects how they learn differently. And so this is really interesting. We feel that this does work, but now we need to understand how, why, when, and all those things in between, so that we can bring this full circle and make sure we can help people that have disorders of memory. And with that, I'd like to thank my lab, everyone in the lab that's helped with this, as well as the mice that made this possible. And I'd be happy to take any questions.
Awesome, Rick. Thanks. That's really interesting. And we got some really good questions going in the chat. So I think we'll start, because this is kind of where you left off, is how would you apply this work to humans? And could you do this in humans?
This is a great point. And I actually meant to bring this up when I was presenting this, but you actually, the optogenetics tool is not really designed for putting this in humans. This idea is more of a way to understand how the brain works normally, because mouse brains and human brains work really similarly. If we can turn those dials in the brain, especially of the mouse brain, and see how it works, that'll give us information about how it works in the human brain. So this really isn't designed to work in humans, but it's more so a tool for us to understand how the brain works. That's a great question.
And then along the same kind of lines, how does this work to increase neurotransmitters? How do these lights work? And do you have to deal with things like bioluminescence and naturally occurring light releasing and things?
Okay, so I'll touch on the first part of that, or the last part of that first, actually. So the bioluminescence and naturally occurring light release doesn't really happen in the brain so much. But since the brain is a tissue, it's like sort of different parts of the body living together, you have to think about how to deliver light through tissue. And so that's why we have to use lasers, because we need strong power. And the way that this actually works is a little beyond me, because I'm not in physics, but I did learn this in neurobiology. So normally, neurons actually communicate using electricity. And so what's really cool about this opto tool is that it's an ion channel. And you'll learn about this more whenever you take AP biology or biology honors. These ion channels are positioned all over this cell, from its cell body down to its arm as well. And so these ion channels are special because they open up whenever you shine a laser at them. And they're actually taken from algae. So it was a really cool way that different scientists from across the fields looked at something that was found in algae and figured out a way to apply it to neuroscience. So there are some cool videos that explain that a lot better with visuals, because it's a really graphic way to sort of understand it. That's a great question.
Yeah, we have a lot of really great questions going on here about kind of different things you could do with this experiment. So maybe just generally, how long does it take you to come up with something like this? And how How long have you been working on a project like this?
Oh, wow. So that's a very good question. So interestingly enough, and I think this is really important, I tried doing this experiment for about a year and a half and nothing worked. Not a single time did it work. And I'm not showing a lot of the data here because I actually had to design my experiment in a way that was just hard enough for the mice to kind of struggle and not get it right away, but not too hard to where they gave up and they wouldn't nose poke anymore. So that took a little bit of time. And also, I had to get really good at the brain surgeries I did because if you miss this little area, then it's not going to work for you. I have to hit this injection correctly, and I also have to plant that fiber above the VLA on both sides of the brain well. So it takes, after you inject the tool, it takes about 2 to 3 weeks. And then I actually run behavior with these mice every day at 7 a.m. for about a month. So it takes a long time to get this data, but it ends up being worth it because I found this stuff really, really fascinating.
Yeah, that's really good and takes a lot of technique. I know I've tried a surgery before too, and it's not easy. So Rick is very skilled.
All right. Hey, everybody. Welcome back. I hope you had productive and fun breakout room meetings. Our next speaker is Chris Anderson, and he's going to be telling you all some really cool stuff involving engineering tissues for helping us with medical applications. So with that, go ahead and tell us about it, Chris.
All right. So thanks, everyone, for coming to today's Exploring Science session. And now I get to talk to you a little bit about my personal favorite aspect of biology. So using stem cell technology to usher in the so-called personalized medicine revolution. So before we get into the topic at hand, I thought I can give you a little bit of my background and kind of my journey into science. So right now I am in my 4th year in my PhD program through the Department of Pathology here at Yale. And I'm also affiliated with the Physical and Engineering Biology program, which just means that I'm able to then use different strategies from the engineering, disciplines in biological systems to try to figure out how can we make things to help patients who might be sick. So growing up, I guess I always had like a mild interest in science, but then just through this kind of chance encounter where in my 3rd year of high school, so junior year, I ended up taking a class where the teacher happens to be a former PhD scientist himself and had this pet project where he would work with these certain plants that had these very colorful, large leaves. And what he wanted to do was he wanted to be able to start making new kinds of patterns by using this chemical called EMS, where you can make random mutations in this plant's genome. And then by doing selective breeding of those plants, you can get all kinds of crazy combination like leaf patterns that just didn't exist in nature. And to me, I thought this was very cool because it literally took all of the different things that we were learning, in the classroom about genetics, about heredity, and applying them to actually do something that, was very real and right in front of you. And then I realized that I could, do that myself. And this all happened just because I was taking advantage of an opportunity that was presented to me, just like you guys are all doing now by joining us for some of these talks. So you're already on the right path to starting your own careers in science. And maybe in a few years, you guys will be the ones on this side giving lectures to the next generation. So what I wanted to talk to you guys about today was, the concept of the personalized medicine revolution. So before we can get into personalized medicine, I wanted to talk a little bit about, the way that modern medicine has been working up until now, and then what we hope to improve upon with this new idea. So modern medicine has really helped to improve the quality and the length of life for people all around the world. And then in the chat, all I want you guys to do is think, so before we started, having what's known as modern medicine, when we started to look at what are the causes of disease and how can we treat it, how long do you think the average person was able to live for? A lot of good answers. So I'm seeing the range from 30 to, let's say, 50-ish years. And it's actually really close. So actually the average life expectancy for most of recorded human history actually was 25 to 30 years. And me personally, as someone who just turned 28 last month, I am very happy that this is no longer the case. But then after we started to figure out, through biology, kind of like Rick is doing, how different aspects of our body work and how we can interact with them, we were able to figure out how we can treat diseases in a much more direct way. So in 2019, the World Health Organization did a study where they determined that now, with modern medicine as it is, the global life expectancy has actually increased to about 72 years. So just by looking at how our body works and understanding it, through the lens of science, we've been able to double the life expectancy of the average person, which is really great. So how have we been doing that? So we essentially, physicians and scientists alike have been using this one-size-fits-all kind of mode of treating people. And essentially what that is trying to use our shared biology, because we as humans all have, we evolved in very similar patterns. We want to look at what's common among us and figure out how can we make chemicals that will interact with proteins that we all, share, and try to treat different issues that we might have with disease. So here we have a picture of aspirin, which I'm sure a lot of you guys have just seen around the house, and you can use that if you're having a fever or if you're experiencing pain and things like that. And here's what aspirin actually looks like at the chemical level. So this is just essentially a cartoon that shows chemists who are working with this molecule how all of the different atoms are arranged in space and connected to each other. And then this, geometry of the molecule is actually what allows it to work with some of the proteins that we all share to influence how we experience, things like inflammation, which has a lot to do with how your fever will, end up progressing, or how we experience pain and how all of that goes. And the main thing that both physicians and scientists are now thinking is, are we all exactly the same? Like, will this one size fits all kind of mode really work for the whole population? And there actually have been studies into that. So right here, I have an actual graph that was taken from a recent study looking at the effectiveness of some of these small molecule therapies for different diseases. And what you can essentially think of is each of these dots is a different therapy that we use to treat a different disease. And essentially, the farther that you go to the right on this graph, the larger percentage of the overall population this molecule is able to help. And then the further you go to the left, the smaller percentage of the population is able to help. And there are a lot of molecules that are able to help a lot of people, but then for certain diseases, you actually have this one size fits all kind of mode of treating patients just kind of break down. You have very few people who respond well to the therapy, and you need to start thinking kind of outside the box, like how do we help the rest of the people who aren't being helped by these simple molecules. So realizing that we're all, different, we're all unique, this is where the idea of personalized medicine kind of came out of, right? So the goal of personalized medicine is to essentially, instead of making one thing that you want everyone to take who has a certain disease, now we want to work backwards and then look at the individual patient, figure out what specifically is going wrong in their body and tailor the medicine to them. And there are a lot of different ways to do that, but the way that we're going to talk about today is known as tissue engineering. So for tissue engineering, you essentially need two main components. So you need the cells that will make up your tissue, and then you will also need the backbone that will organize the cells inside of like a three-dimensional space, right? So first, let's talk about where are we going to get the cells to make a tissue, because you need a lot of them. And where tissue engineers generally go for their cell sources are these cells called stem cells. And the definition of a stem cell is really just any cell that is able to turn into other cells in the body. So here are some pictures talking about how we as tissue engineers are able to go about and get our stem cells. And there's two main ways that we're able to do that. So the traditional way to get a stem cell is where you can take what's known as a zygote. So this is essentially a recently fertilized egg that has DNA from both the mother and the father. And then very early on in its cell division, it will form this structure known as a blastocyst, where these cells on the inside of this structure can be harvested, and then they're what's known as pluripotent stem cells, where the definition of pluripotent stem cells is just that they can actually replicate themselves indefinitely. So you can have theoretically unlimited numbers of cells for whatever tissue that you want to make. But then more importantly, they're able to turn into all cell types that make up your body. So they can turn into neurons, like we were just talking about in Rick's presentation. You can get lung cells, you can get cells of your pancreas, you can get smooth muscle cells, red blood cells, the cells that carry oxygen in your blood, or you can also get heart muscle cells, which is what we'll talk about a lot about today with my research specifically. But more recently, so actually just back in 2009, a group out of Japan actually figured out how we can take cells from an adult patient and reprogram them to act like stem cells and have all of these same capabilities. And this is just a very simplified picture of how we do that. So we essentially take the adult cells from the patient, and then we can introduce these 4 genes into the cell that will then reprogram it to act like a stem cell. So then with that, now we have this same unlimited cell source, but it's now going to be recognized by the patient that you're making this for as their own tissue, which is great. So now you don't have to do any kinds of extra things to get their body to accept it, which is really great for making this personalized therapy for them. So now that we have our cell source, we want to start talking about the engineering of things. How do we now take our cells and make something that we can put into a person? So that's where the field of tissue engineering comes in, which is broadly defined as a field of science that actually combines a lot of other fields of science. So you take into account physics, different concepts from engineering and biology to get all these things to work together to make something that will be both safe and function for your patient. So here we have a bunch of examples of different engineered tissues, and I want to give everyone a chance in the chat to kind of guess what you think these tissues are. I've seen a lot of good answers already, yeah. So you guys know your body parts. So we can go down the list really quickly. So some of you got it right away with A. A is actually skin. So this is for people who might have gotten bad burns and need a skin graft to help cover up that tissue that's missing. B is actually 3D printed bone, and I saw a lot of people got that as well. C is a heart valve. So this is one of the valves that will control how blood moves through your 4 chambers of your heart. D is an engineered bladder. E, a lot of you got this as well. So this is actually engineered liver. So that's very great. And F was tricky. So F is actually a urethra. So this is the vessel that will carry out your liquid excrement. So your liquid waste if you drink a lot of water, which you definitely should because it's getting very hot nowadays. So great. So how do we actually make these tissues? So here's a snapshot of the type of work that my lab at Yale does. So we are a cardiovascular engineering lab. So that's just a big fancy word meaning that we make tissues of the heart and circulatory system. And then we can do that for either looking at how diseases are caused in human tissue, or what I do, try to make tissues to treat different types of patients. So just like we talked about before, we can take a sample of the patient's cells and then reprogram them to act like stem cells and start making different cell types of the cardiovascular system. So we have cells of blood vessels that we can use to make engineered vessels for either therapies or small engineered vessel rings that we can use to look for disease progression in the lab. And then I work on the heart side of things, where we can take heart muscle and then use different strategies to make heart tissues to look at different diseases, to test different small molecules and actually see how they will react to human tissue and how that affects function. Or what I do is I can actually take formed like protein backbone from other organs or other animals, wash away those animal cells, and then put in the patient's human heart muscle cells to make tissue that is ready to go into a person. So here's actually a video of some of my heart cells after they go through that differentiation process. So basically going from the stem cell into a heart cell, and you can see that they're actually beating on their own. So without any kind of cues from me, they're contracting, they're moving, they're doing mechanical work, and they are ripe and ready to start making our tissues to make these therapies. But as you can see, like these cells are very disorganized. They're not really in any kind of structure. So this is why we need that backbone to organize them into something that will have all this work be used in a very specific way. So how do we now that we have the cells, how do we organize them? And a very popular method in tissue engineering is what's known as natural scaffold strategies. So essentially using, the naturally occurring protein backbone of organs from other, from other animals to organize our human cells. So a very popular method for doing this is what's called whole organ decellularization. And that's just a fancy word meaning taking out all of the cells that are already in the organ, so that way it's all ready and primed to accept our human cells. So what we're able to do is we can actually take a fully formed organ from an animal that has very similar, structure and, mechanical properties to us. So usually pig, they have a very similar structure to human hearts. Put them inside of the special rig known as a bioreactor. And then we can actually flow through different chemicals of all 4 chambers of the heart. And that's going to wash away all of the pig cells. And once that process is done, you end up with this, what's sometimes called the ghost heart. And it's all very opaque and almost, see-through, because it's just that protein backbone. But the cool thing is it still has all of that 3-dimensional geometry that we need to organize our own cells. So here's an image of what that looks like through time. So you can see here where we're just starting the process of taking away all of the native cells. It still looks very red, very muscle-like. And then just as it keeps going through the process, it eventually gets to that ghost stage, right? But then what do we do with it after we get that protein backbone? We can then use the same type of system to then flow our engineered human heart muscle cells through those chambers of the heart and then provide electrical stimulation, because the heart takes electrical cues to help the cells contract and organize to one another. And essentially what happens through there is you can go through the decellularization process, get this ghost heart, and then start to flow through the human cells, and they start to organize, and you can see it's getting its color back and starting to look more like a fully formed heart that you would find inside of a person. And then, unfortunately, I couldn't find the video for you guys, but actually this came out of a group in Texas Heart Institute down in Houston, where they were actually able to take the heart at this stage and then electrically stimulate it. You can actually see the different chambers beating with one another. So essentially, they're able to actually make an organ that would respond to electrical cues and do mechanical work, which is really important for heart tissue. And then this same kind of approach can be applied to a lot of different organs. So right here at Yale, we have Dr. Laura Nicholson, who's doing the same kind of thing, but with lung tissue. So she's taking lungs and then washing away all the cells that are already there, and then using the same kind of technology to flow cells through the different structures of that lung and then repopulate it with the human patient cells. And then another thing that a lot of bioengineers are doing now is trying to use 3D printing to make whole organs just from scratch. So just raise of hands, like who has ever seen, or who's heard of 3D printing before, or ever seen a 3D printer in like action? Yeah, so it sounds like a lot of you have, right? So as I'm sure a lot of you know, 3D printing is literally just printing in 3 dimensions. So not just the 2 dimensions of a sheet of paper, but then doing things layer by layer where you are going up in that 3rd dimension to make something that you can then take and be, you know, this really cool thing that you can actually hold. So tissue engineers are trying to adapt this technology to then work with cells and these different chemical compounds known as bioinks. So essentially all a bioink or cell ink is it is, you can actually think of it almost like really strong jello, where you can mix your cells that you got from your stem cell sources into this jello, and then as you're printing it, you can then use light to turn it from a liquid into a solid. And then as you go about doing that, you can make really complex and cool structures. So here we actually have different heart chambers being 3D printed. So this is a heart chamber known as the ventricle. So this is where you actually have heart muscle cells kind of layered all throughout this tissue, and then they will respond to electrical stimuli. But then very recently, there was a group that was actually able to use what's known as immersion 3D printing. So just actually using this liquid bath to kind of help keep the structure in place while you're printing it, so you can do a lot more complex 3D prints. And they were able to print an entire heart that had different cell sources. So the vessel components had vessel cells, the muscle components had muscle cells, and they were actually able to pump it with different fluids that were held in place and all in the right chambers. So that is where the technology seems to be going, where hopefully one day we can actually start printing organs kind of on demand for people who need transplants. But through then stem cell technology, they will be the patient's own cells. So in conclusion, tissue engineering is a really cool new tool that allows us to start using nature as a starting point and figuring out how do we in the lab kind of recreate that and make organs or smaller tissues that we can implant into people who need, you know, these kinds of replacement therapies. And then through stem cell technology, we're able to then start tailoring it to the individual, where these organs are designed to be recognized as your own tissue, your own cells. So they'll hopefully work with your system a lot better. And with that, I can take any questions that you guys might have.
Great talk, Chris. Seems everybody was really excited by that, but there were also a whole bunch of ews throughout it, which I think is a pretty normal response. We'll do a couple questions here, and we won't be able to get to all of them, but we will be doing sort of a follow-up interview thing with Chris afterwards that we will be posting. So if your question doesn't get answered here, look for that in the follow-up thing that we'll send out. But for you, Chris, one question we had was, how long do the decellularization and recellularization processes take?
So the decellularization process actually takes about 2 weeks. So it is a bit of a long process, but the cool thing about it is once you get rid of all the cells, you can actually store that ghost tissue or that backbone in just a normal fridge, just as long as you have it submerged in a liquid, and it will last for months. So what a lot of researchers are doing are trying to design their tissues to get to that stage, and then you can just easily store and ship them just like you would any kind of meat that we have in the grocery store, right? Have them shipped to hospitals across the country, and then hospitals, as they need them, can take them off the shelf and start getting them ready for patients as they need.
Yeah, really cool. And then maybe going into a little bit more detail on this, if you take away all of the cells from the heart or whatever other organ you're going to decellularize, what's the thing that's left over?
Yeah, so the thing that's left over is actually just proteins that your cells put outside of their own, their own membrane. So all of your tissues are actually not just cells connected with one another, but they actually have what's called this extracellular matrix, which is just a bunch of proteins that can help them, maintain their structure while they're doing whatever function that they need to. And it's really important in the heart because you can essentially think of the heart as just this actual pump that needs to do a lot of physical work, that needs to do a lot of moving. So it really needs this protein backbone in order to just not fall apart while it's doing what it needs to do for pumps.
Fantastic. And then as one last question, there were a few people that were getting at the idea of just how effective are the 3D printed organs that have been made so far?
So 3D printing, it depends on the type of organ and the size. So a really interesting TED talk that was coming out like maybe 5 years ago was actually looking at 3D printing kidneys, where they were able to print an entire human kidney, but it took about, I think, like 30 hours. And essentially, by the time that they were done printing the kidney, the cells on the inside of the kidney, because they couldn't get nutrients, were already dead. So there is this trade-off between how long it takes versus how to keep the tissue alive. And with what we were just talking about, once again, with the heart, which has a lot of physics that are really important for it to be able to function. I know I showed you an image of a 3D printed heart that had, the really complex structure and the different cell types, but it didn't have the mechanical properties to be able to actually beat and move blood like you would need from your native heart. So essentially 3D printing is kind of finding the balance between those with speed, keeping the cells alive, and also getting the mechanics right for whatever function you want the tissue to have.
Fantastic. Well, thanks a lot, Chris. This was a great talk, and I think people have been very excited to hear it from you. We have our two speakers from the May 26th Exploring Science event here. We have Rick, who presented his Flip Science Fair poster, and Chris, who presented his Science Cafe talk. They were both received very well. We had a lot of questions and a lot of engagement, and I think students will be very excited to hear even more answers from you both. So we'll start off with Rick, and we had a number of students who were interested in how the mice in your experiments, what their life is like. Does it hurt for them to get introduced to this rig, and what happens after your experiments are done?
Yes, that's a great question. And as I started my talk as well, talking about animal research, I wanted to make sure that everyone understood that this is an extreme privilege for us to be able to work with animals, and veterinarians have to approve all of our experiments. And so whenever we do surgeries with these mice, they're actually under anesthesia. So like if you went to the dentist and got a tooth pulled out, they're under a sleeping gas. And we also give them pain meds afterwards. And so to get them used to the rig, for your question on that, so I actually, a week before I start putting them in the box, I just hold the mouse in my lap for 3 minutes a day, each mouse for 7 days, to get them used to me so they're not stressed out. And so they actually just walk right into your hand afterwards, because they love hanging out with you, basically, because they know they're going to get milkshakes. So their lives in that sense are pretty cool. They seem to like it. They don't bite you anymore after you handle them. And, they have the optic fibers sticking out of their head, but for them, I've gotten really good at surgeries and making sure there's no infections. And so it's kind of just like a top hat for them. They don't really seem to mind it too much, and you can connect the lasers to it and they run around and it's fine because it's on a swivel. But one of the saddest parts that I find the experiments is at the end, we do have to sacrifice the mice and take their brains, because I've done all these really complex manipulations where I inject somewhere in the brain and then I put an optic fiber in the brain. And to really understand if that data was actually doing what I thought it was, we have to look at the brain and make sure I hit the target injection site right and the optic fibers were in the right place. So I do 2 injections and 2 fibers. So there's a lot of chances that things can go wrong. And so you'd have to exclude that data. So we have to put the mice to sleep for that. But we do that sort of what you might do at a veterinarian office for an old dog who is suffering or something like that. So they don't feel it and they go to sleep. Yeah, so it's a sad thing, but we take it very seriously. We really appreciate their sacrifice.
You get some really good and important answers out in the end too. Very important things that you learn by being able to work with these mice.
Absolutely, because the brain is so complicated. Some people will say, well, maybe you can do brain cells, like grow them outside of the brain. But if we understood the brain well enough to recreate the brain in order to study it, then we wouldn't have to study it at all, because we'd already have it all figured out. So nature gives us these models, this way to study the brain in a more simple system. But yeah, some of these questions just can't be gotten at. I can't teach a petri dish of brain cells how to nose poke for milkshakes. Cells don't really like milkshakes.
But on the note of milkshakes, there was a lot of comments about it, and there were questions too. And one kind of cool one was, for the mice that do get to have the milkshakes, presuming that gives them some excess energy, does that excess energy potentially affect your results at all, do you think?
So that's a really fantastic question, because this was actually a question in one of my breakout rooms too. And so the milkshakes that I'm giving them are not just strawberry milkshakes. These are actually protein nutritional milkshakes. So they have high fat content, high protein, and they're actually really healthy for you. They're Ensure, which is what you might have if you're sick, or maybe your grandparents drink that whenever they're getting elderly. And so that's a fantastic question. Are these mice just sort of amped up because they have a lot of sugar and they're ready to nose poke more and get more? Do they have more energy? And we actually do look at that. So we looked at the number of errors that those mice made as well, both groups. And actually the mice who are not earning as many milkshakes, for instance, the controlled mice took about 8 days to learn. So if you look at day 4, they're still making a ton of nose pokes. They're just making incorrect ones. So it's not like they're tired because they're not getting milkshakes, but they're just not able to learn the fact that they need to only nose poke when the feed comes on. So we have a control built into that experiment, but it's a really fantastic question.
Great. That's really cool to think about. I think then this last one was a big sort of underlying question, sort of getting at what has to be considered one of the coolest features of like how you're able to perform this experiment. But there are questions about how actually do you use the laser to change the amount of neurotransmitter that's in the brain?
Yeah, so this technique, optogenetics, is actually what inspired me to do neuroscience. So I was working as a biologist in a cancer drug company for a while, and I was listening to NPR, and I heard them talk about how they were reactivating certain memories by just using a laser to turn on brain cells. And so I looked into this more, and I mentioned this a little bit in the Q&A, but this molecule is called channel rhodopsin, and it's what underlies the optogenetic tool. It's a protein that is an ion channel. And so if I was a brain cell and I was sending my arms out to release acetylcholine or any neurotransmitter, this is my arm, and on the membrane of it, there's going to be these ion channels that I put on that are the opto tool. And so normally your brain cell has these ion channels throughout, and this is sort of how it uses electrical transmission to release its neurotransmitters to get from the cell body to the end of it where it releases those chemicals. But these are sort of extra ion channels that open up, instead of with electricity, with light. And so you're able to shine a laser and cause them to sort of hijack the system and open up the floodgates to release neurotransmitters. And so we're not really making these neurons make more neurotransmitter. Maybe secondarily we are because they're releasing more. But you can imagine that at the tip of my fingers, I have these neurotransmitters ready to go. And so the light opens up those ion channels and then causes a process that will make the neuron release more from it. So it's sort of just like, honestly, flipping a light switch onto the neuron. everything in your house is already set up to turn on the electricity and make the lights turn on in your house. But we're just sort of going over there and turning the light switch on by shooting a laser at it, basically. So it's a fantastic question, and it's actually extremely complicated. So it was really cool. A lot of physicists and molecular biologists worked to make sure that this could go into neurons and neurons could actually do this. And so, so I think it's a really fascinating story. And there's some TED Talks on it that show you actually in an animated way what's going on.
And correct me if I'm wrong, but the rhodopsin part of that ion channel name, rhodopsin is also a pigment molecule that helps your eyes work, right?
Yeah, that's right. And so these are, this is the way sort of the basis for your eyes capturing light photons and telling your brain what you're seeing. These ion channels are in the light as well as algae.
Cool.
Or are in the eye as well as algae.
Well, thanks a lot, Rick. It's a really cool talk, and I think the students will appreciate hearing more from you.
Thank you.
And then, yeah.
All right, Chris. So many questions for your talk. Fantastic. I think, yeah, people are very interested, a little bit grossed out, but I think everyone kept their lunch down, it seems like. So, okay, we just have like a couple extra follow-up questions for you. First of all, you talked at the beginning of your talk a lot about how you guys are using stem cells, but you're not using like the stem cells that stay as stem cells, but ones that you can kind of create. So how does that process work?
Yeah, so essentially by studying the old stem cells, what you would get from, what's known as a zygote, we can figure out what genes are turned on and off in those, those stem cells that you would get, I guess way back in the day. And essentially what we're able to do is by using those 4 genes that I talked about, you actually are able to change the gene expression that you have in the adult cells that you can get from the patient. So there is this concept, very broadly speaking, known as epigenetics. So how every cell in your body is expressing different genes based on the chemical modifications to your genome or all the DNA that the cell has. And essentially what those 4 genes that we call the reprogramming factors to turn your adult cells back into stem cells, they're changing the way that your genome or your DNA is chemically modified. So that way the cells that are active in that stem cell state are now the ones being expressed, and all of the ones that are useful for the cell types of your adult or growing body are now turned off, and you have this blank slate that you can then start to push into whatever cell type that you need.
Fantastic. That was a great explanation. Thank you. Another question we had is, so these organs that you make using the stem cells through recellularization, would these organs be able to grow with your body? Like if you got this as a kid and you're still growing, would these organs be able to grow?
Yes, so that's actually one of the major goals for tissue engineering is you want to have your tissue that you make in the lab behave just like the tissue that would be made in, your body, right? So with either the 3D printing or the natural scaffold strategies, all these engineered structures would be made of different chemicals and molecules that you would naturally find in your body. So because of that, your cells know how to interact with them and then how to move them around to make them grow with, you as a patient. So a lot of these, so actually even specifically my research is actually trying to engineer a tissue that we would want to implant into a newborn baby because they have a heart that didn't form correctly. So the main goal with the tissue that I'm making is actually to have it integrate with the rest of your body. So that way, as you go through and you're having your growth spurts and, gaining a lot of, height, your tissue that we implanted is going to grow in synchrony and all at the same time.
Very cool. Okay. We had some questions about engineering that you can do with different organs. So you talk a lot about the heart, but what about your eyes? So can you help someone who has color blindness maybe? Could you add like new features, like add some infrared vision to these eyes? Any thoughts on that?
Yeah, so that's actually, yeah, with something that we can do. So since we are making these tissues in the lab, we can control what aspects of the tissue that we want to put in there. So for instance, like if someone's colorblind, and let's say that happens because in your eye you have these structures called photoreceptors. So the actual proteins that will respond to light of different colors or wavelengths that will then be processed as information that your brain will then make an image out of. And let's say that you're missing some of those photoreceptors that can let you see certain colors. We can definitely, as we're making this tissue in the lab, add those back. But then going further than that, and this gets into the concept of this realm of science called synthetic biology. So actually using what we know about biology and physiology to then make things that don't exist in nature. So if you wanted to, let's say, be able to see ultraviolet or infrared, so things are normally outside of the spectrum of light that humans can naturally see. If you wanted to make an eye that had engineered receptors that could see that, that is something that we could do at the engineering level once we have the technology worked out.
Super cool. Okay, so kind of going along those lines of what you're talking about, making like new things, could you make a whole new organism using this? Like, if we can make a heart, and you said we can make like a liver and lungs, could we put this all together and have a whole new, maybe a human, a clone? What are your thoughts on that? Maybe the ethics and the science side.
So cloning, yeah. So theoretically, once we figure out how to make all of the different tissues of the body, we actually could. Obviously, before we get into the ethics, there are probably easier ways to clone organisms. So the best example of that, I would say go look up Dolly the sheep, I think it was, which was one of the first clones that was made using genetic engineering technology. But then the ethics of that would be very complicated in terms of, if we're making a new person, like what do we start thinking about in terms of consent with not just the individual that's cloned from, but then, what about the rights of the person who is, the clone. And there are a lot of actual, I guess, movies that do touch on these subjects that I guess maybe once you ask your parents and they think that you would be old enough to watch them, things like I think Blade Runner is an old school movie from like, I don't know, like the 80s, sometime way back, that touches on this kind of concept. And then Altered Carbon is one that actually does that exactly with 3D printing whole bodies. And it's really interesting to look at not just the science of can we do it, but then another important aspect of any new technology is figuring out should we do it, and how should we do it, so that we're making sure that we're not being unethical in our actions.
Fantastic. Yeah, lots of things to think about. Even if we can do it, should we? It's a great kind of thing to always have in mind. Okay, well, thank you both for staying after to answer all these questions. We really appreciate it.
Yeah, thanks for having us.
Thanks.